So today we'll be talking about a new topic which includes the one method which is involved in the recombination of bacteria. How can bacterial genes recombine and hence one property of a bacteria can be transmitted to the other bacteria through the means of plasmid. So what are plasmids? So plasmids are basically extra chromosomal pieces of DNA which are not the part of the normal chromosome of a bacterial cell, and they contain a specific function. So, first of all, plasmids were considered to be accessory parts of the cells, as in they were not required for the survival of the and reproduction of the cells. But now it is proved that plasmids confer antibiotic resistance to bacterial cells. So basically they have the resistance genes on them which can help the bacteria resist the dangerous antibiotics for them. Which is why nowadays there are many bacteria which are not treated easily upon antibiotic intake. Okay, so this was a basic overview of the plasmids. Now we're going to be discussing about uh, the genetic recombination which takes place between two bacterial cells, in which one bacterial cell transfers its plasmid to the other cell with which it is connected. So basically, as you can basically catch that this process is the conjugation. Okay, so what is conjugation basically? Sorry. So conjugation is a genetic, let me just decrease the size a bit, recombination method where genetic material is, in most cases it is a plasmid. It can also be the normal chromosomal DNA, are transferred between transferred from one bacterial cell, so you all know I'm talking about the bacterial cell, to other cell via direct contact. So this is basically a very important point which distinguishes conjugation from other genetic recombination processes occurring in the bacterial cells. Other genetic recombination methods do not require the two bacteria to be in a direct contact, but the conjugation process is a bit different, in which two bacterial cells are directly connected to each other with the aid of sex pilus, which is also a property of the plasmids. The cells which transfer their plasmid to another bacterial cell also form the sex pilus. They also have the genes that code for sex pilus. Okay, so in easy words I'll be explaining it, don't worry. So just suppose that this is a bacterial cell. Okay, let me just alter it a bit so that I can. Okay, this is a bacterial cell and this is a bridge. This is a sex pilus, basically sex pilus. Okay, and what is happening with this sex pilus? Usually, yeah, This is another bacterial cell associated with the sex pylus. So you can say that this is the cell. I'm gonna show the plasmid with the purple color, okay? So just and the normal chromosome with this yellow one, okay? So this is the normal one. It also contains the one, okay? So purple one is the plasmid, okay? So this one has a plasmid, but what will happen? This cell is in a direct contact. It forms basically a sex pilus, which extends to another cell, and these two are in a direct contact ready for the conjugation method. So basically, this plasmid will travel all the way here to here as well. I'm just giving a basic idea. I'll explain step by step, okay? Don't worry. So this is the plasmid which has been transferred from one bacterial cell to the other, and this can also be called as the mating bacteria. This is basically a mating type. Conjugation is basically mating between two bacteria. So these are also mating bacteria, okay? Okay. So now I just wanted to go a bit into detail so that you all can get the basic overview that what is happening, what is conjugation, so it will be easier for you. I'll be explaining other genetic recombination methods occurring in bacteria in other videos, so please do watch them whenever I upload them because it will be really helpful for you in terms of microbiology. Okay, so now what causes, so basically there's a type of plasmid called plasmids which are called the conjugative plasmid, okay? So this conjugation process is basically, the plasmids are literally like they're carrying out the whole process. They are causing the sex pilus to extend out. They have the plasmids. They are transferring the plasmids from one cell to another cell. These are the conjugation bacteria plasmids. Sorry, conjugation plasmids. Okay, so their function another function is that they contain genes for sex pilus, and the other one is that they are transferred from one cell, sorry, bacterial cell, because we're looking at the bacterial cells, okay? Okay, so just note that there are other types of plasmids as well. These are one of the plasmids which are specifically made for this function, for the conjugation process, okay? So now I'll be discussing the process in detail. Okay, so there are two types of cells. Yeah, we are looking at the bacterial cells, but one of the cell is a donor cell, okay? And additionally, it is also called F plus factor. Basically, it stands for fertility factor, okay? So this plasmid has the property of fertility factor, and it's gonna um, transfer its plasmid to another cell with F minus factor, meaning that it doesn't have any fertility factor or any plasmid. The plasmid is basically the F plus factor, okay? F factor basically. Okay, and there's a recipient cell. Let me use the same color. There's a recipient cell, which is the F minus cell, okay? Or factor. It's the same. Okay, so what will happen in the most simple type of conjugation method? There are many types of the there are many possibilities of how the process will progress. One of them is the simple when, so I'm going to be drawing a cell again here. So, okay, so this is a donor cell, okay, and it is forming a sex virus, okay, with the another cell with no plasmid. Sorry, that's not nice. So one of these cells has the plasmid while the other lacks it. So I'll be showing the normal chromosome with a purple color. Okay, these are the normal chromosomes. And now the plasmid one is here. This is the plasmid in only the donor cell and not the um this recipient cell. That's how it makes it this one a donor cell, okay? This is the donor cell and this is the recipient cell. Okay, so what is happening here? So what will happen when the sex pilus is extended by the F plus factor? Okay, I'll be writing this as well for so that it's easier for you all. This is the F minus cell lacking the plasmid with the F plus factor. Okay, okay. So what will happen when these two come in close contact? What will happen next is that, so I'll be drawing again this one here. So I'll be just increasing the size just to show you all the, Okay, so this is the normal chromosome and this is the plasmid one. What is happening? As the DNA is double stranded, one strand of this plasmid, one strand only of this plasmid will go into this cell with the aid of this sex pilus, and thus it will also contain this one strand of the plasmid. Now, what will happen next? That these two will itself, these two single strands of DNA will itself replicate and completing thus the double-stranded DNA molecule in these both. So what is happening? This one was an F cell and it is still an F cell. This is a very important point that these are still the F plus as they didn't like transfer didn't only transfer their plasmid but they also have that plasmid, so these are still the F plus cell. This is still the F plus cell and this one has been mutated to an F plus cell. This one is the recombinant F plus cell. Okay, so this was one situation in which conjugate plasmids can be transferred from an F plus factor to an F minus factor. Okay, so this is the one of the cases. Now next I'll be discussing another type. So let me just erase these all. Erase these all. Okay, so let me just rewrite it here that we're discussing the conjugation method. Okay. Okay. So next situation is a situation where, so this is again an F plus cell with the normal chromosome and the F plus F factor. Okay. So this is an F plus cell or factor. Okay. So what will happen is that little plasmid can this little plasmid can go over to its own chromosome, and it can recombine with the chromosome, with its normal chromosome. Okay, so what did I just say? I said that this plasmid will recombine with its own chromosome, its normal chromosome, inside the same cell. There's not conjugation process going on right here. It's still more. Okay, so So this is just a basic process happening inside its cell. It's the basic recombination event going on, but there this is not the conjugation process, okay? Not conjugation yet, okay? This is an important point, not conjugation yet, okay? So what it okay. So this is the same. I'm sorry, this is a bit messy. Let me just make an oval shape, I guess. Okay, this is a bacterial cell, okay? So what did I just say? Oh, sorry. Okay, so this is the normal chromosome, and imagine that there's a, let me just make it a bit bigger. There's a small piece of plasmid DNA which has combined with the normal chromosome of the cell. Is it like distinguishable? Let me just highlight it a bit to be more clear. Okay, this one is the plasmid one, which has recombined with its own DNA, with its own chromosome in the same cell. Okay, similarly, let me just write it here that recombination between plasmid and chromosome DNA in same cell, same bacterial cell basically. Okay, so this was initially an F plus cell. Now what is now this will be called an HFR cell. Okay, it basically stands for high frequency recombination, as this is a really high type of recombination. So this is called an HFR cell, not the F plus cell. Just keep in mind that F plus cell has separate chromosomal DNA and separate plasmid, but the HFR cell has the recombined chromosomal DNA and the plasmid. Okay, so this is the plasmid. Just let me write it. While this is the chromosomal DNA. Okay, and as you will know that bacterial cells have a single circular chromosome, okay, and DNA as well. So that's why these are in a circular form. Okay, so this has become an HFR cell. Okay, great. Now what will happen next? This, just keep in mind that this is a basic recombination event. This is not a conjugation process yet. There is still one more case in the conjugation process, and it's linked with this HFR cell. Okay, so what will happen next is that this not normal HFR cell has its normal chromosomal DNA combined with the plasmid. Oh, I'm sorry, this is a bit messy. Okay, so this is a normal chromosome, and this is the plasmid which has been recombined. Let me just put a dark color so it's more clear because it's not distinguishable. Let me do a blue color to the plasmid, okay? So let me write it here that blue is the plasmid and purple is the chromosomal DNA. Okay. Okay, so let me just, yeah. Okay, so this one is the HFR cell. Okay, so now we're going to be discussing about the conjugation process. which will be occurring between an HFR cell and an F minus cell, not F plus cell, F minus cell, okay? So this is a 6 bias, okay, and this is another, let me do a dark color, and this is another bacterial cell, okay? So now what will happen? This is the 6 pilus, okay? Okay, so what will happen? So what will happen is that basically this part, this chromosome, this whole chromosome will replicate. It will replicate. It will replicate into its another copy, basically. And this, suppose this is the replication. I'm not showing. This is basically replicating, okay? It doesn't have to. It is replicating. And now what will happen that this replicated form will travel all the way to here, and it will now enter the F minus cell. I'm sorry, I forgot to mention F minus cell, okay? So F minus cell only has a normal chromosome. Okay, now what will happen? A portion of this chromosome is going into this cell, okay? So now what will happen? What will happen is that a bit of the chromosome, but not the, let me just put this it's chromosome because it's not distinguishable. What will happen is that this is suppose the chromosome of F minus cell, and this is of F plus cell. Sorry, HFR. Okay, so now what is happening here? So this is the normal bacteria with its chromosome, the F minus cell, and now what will happen when this replicated chromosome plus plasmid are going into this cell? What will happen is that only a portion of the chromosome will be able to integrate into this chromosome. And just there's a huge point to notice here that the plasmid has not been transferred. What is the important point? Plasmid has not been transferred to F minus cell. And remember, only when a plasmid enters the F minus cell, only then will the F minus cell become an F plus cell. Okay, so what is happening in this case is that uh, when this chromosome replicates, a portion of its chromosome goes into the F minus cell, and the portion that has gone into the F minus cell is the normal chromosomal DNA of the HFR cell and not the plasmid. So this cell, what will now the cell be called? This was an original F minus cell, okay? So now what it has become? Now it will be called as recombinant F minus. It will not be called an F plus cell, but it will be called a recombinant F minus cell, as the plasmid has not been transferred from this bacterial cell to this bacterial cell, and only the portion of this normal chromosome has been transferred and integrated into the chromosome of F minus cell. So this is a recombinant F minus cell. So this is a condition, second conjugation method where an HFR donor transfers its replicated recombinant chromosome to an F minus cell, which will result in the formation of a recombinant F minus cell and not the F plus cell. So now let me just go through the advantages of the conjugation process. Why is it important for bacteria to go through conjugation process? And why must plasmids be transferred from one bacterial cell to the other? So basically one of the advantages is that plasmids often contain important genes for the survival of a bacteria if conditions of the environment change. When environmental conditions change. So what is meant by this point? Okay, so what is basically the cause is that basically the importance, so we're discussing the advantages. Okay, so basically what is meant by this point is that um or more often times when bacterial cells are often exposed to antibiotics, they are susceptible to that antibiotics. But what there's a twist here that there are some antibiotic resistant bacteria such as the methicillin resistant SRES and many other methicillin resistant, sorry, SRES. So S stands for Staphylococcus aureus. Okay, so these bacteria are methicillin resistant, and they have been there is a really high chance that these plasmids are basically it's not high chance, it's confirmed information that plasmids code for resistance genes which confer antibiotic resistance upon their bacterial cells, okay, upon their hosts. And that is why it is important for these plasmids to be transferred from one bacterial cell to the other bacterial cell, so that those bacterial cells can also be resistant to the same type of antibiotic they are often exposed to. So this is a highly important and most carried out process carried out by the bacterial cells for their genetic recombination. And there is also a second importance of this conjugation process, that it basically increases the diversity in bacterial population. It increases bacterial diversity in their populations, okay. So basically there will be many uh, different and various base sequences in the DNA, as the DNA will be recombined and then combined with the plasmid. So their sequences will be of many types and of many varieties there will be available. So this offers bacterial diversity in their population. And the last one is that this process also helps bacteria survive and reproduce. So, like if the bacterial cells are exposed to a harsh environment, their plasmids must be containing important genes that code for the proteins which can help them in their harsh environments. For example, if the bacterial cells are placed in an environment where there is a higher, so let me just demonstrate it here because it's getting a bit complicated. I'm still on the advantages, by the way. So what I'm saying is that bacterial cells can, if there's a bacteria, for example, this is a bacterial cell, okay, and it is suddenly it has been exposed to into an environment where there is excessive oxygen, okay, excessive oxygen, not, and just suppose that this bacteria is not an aerobic bacteria, it's an anaerobic, just suppose, okay. So I'm explaining the second point which I've discussed, sorry, the protection point, that there are many that how does these plasmids help the bacteria in surviving and reproducing in harsh environments. So just suppose that this bacteria has been placed in a sudden environment where there is a high amount of oxygen, okay, in the environment. And just suppose that this is a bacteria which is an anaerobic bacteria and not the aerobic bacteria, meaning it does not use oxygen for its growth. It cannot grow in the presence of oxygen. So just suppose that this bacteria has a plasmid here, and just suppose that this plasmid has antioxidant genes coding for proteins which are antioxidant, and they which can neutralize the these cells against the oxidant stress, okay. So this is very important for the bacteria to have this plasmid if it is placed in an environment with a higher amount of oxygen, because they will help the cell to reproduce and survive rather than dying. So basically, I'm talking about the advantages, okay, of the plasmids, and conjugation thus, as well as it can also be the cell can also be conjugated to another bacteria, and it can transfer these antioxidant genes to that bacteria again, which will be helpful for that bacteria. So plasmids, so let me write a basic one of the one I'm explaining is that when anaerobic are suddenly placed in a higher oxygen environment, the antioxidant genes in the plasmids will help the bacteria to survive and reproduce in the higher oxygen environment, which would normally cause them to die as their growth will be retarded. Will help the bacteria to reproduce and survive as well, hence, and survive, obviously they'll survive, okay. So this is a highly important advantage of the plasmids, and hence the conjugation process. By the conjugation process, it can be speculated that basically, just suppose that this is a bacterial cell. Okay, let me just quickly go through. I'm not drawing a good one, I know. So just suppose that this is the, okay, maybe I had to make color blue. Okay, so just suppose that this, huh, yeah, just suppose that this cell which has the plasmid for which codes for antioxidant proteins. So this, let me do the orange color. Okay, so this is okay, well, it was the orange one I used. Maybe it was this. Okay, so this is the same plasmid which codes for antioxidant proteins. And this one is the normal chromosome. Okay, this one also has it. And just suppose that these both are anaerobic bacteria anaerobic. So what will happen? This will then go over and one strand will go over here and then these both cells have plasmids with antioxidant genes and hence proteins. So you must also know that genes basically are small regions of DNA which code for the proteins. Okay, this is a very important point. I'll be discussing in my future videos. Okay, so this was all about the conjugation process.